BRCA Gene Final Project by Gabriela and Gustavo Serena. AP Biology has provided an opportunity to study real-life cases such as this one and apply all we have learned to understand the inner workings and science of the body. In this case study, our team focuses on applying all the information we learned throughout the year onto this lab to better understand and memorize each topic or field. Together, we will test ourselves by implementing each unit to identify the BRCA mutation present in individuals and how such cancer occurs. Unit 1, Chemistry of Life. Nucleic acids are a type of macromolecule. There are 4 macromolecules: proteins, lipids, carbohydrates, and nucleic acids. Nucleic acids are made up of CHOMPs, carbons, hydrogens, oxygen, nitrogen, and phosphate. The monomer is nucleotide. The nucleotides consist of a phosphate, nitrogen base, and a deoxyribose. Nitrogen bases are A, T, G, and C. A and T have 2 hydrogen bonds, and G and C have 3 hydrogen bonds. The DNA backbone is made up of phosphate and sugar and is negatively charged and held together by phosphodiester covalent bonds. This relates to the gel electrophoresis, which breaks apart the DNA because there is a negative and positive charge, and the positive charge pulls on the DNA backbone, which is negatively charged, breaking apart, creating varying nucleotide sequences that result, and the smaller sequences move faster to the positive gel side, which results to the varying sizes of DNA within the gel. Unit 2, Cell Structure and Function. The central dogma, DNA to RNA to protein. There are multiple organelles in the cell that have different functions. The nucleus contains DNA, which is the information for the body. The nucleus is semi-permeable and has a double membrane. The molecules provide for the cell, and the molecules get into the cell through passive transport, active transport, and a subpart of that is endocytosis, which is quite important. The BRCA cells are cancerous and uncontrollable, so they duplicate with no checkpoints, and there is no apoptosis. The BRCA cells feed off of resources transmitted into cell, for example, endocytosis, which provides all the food and resources through active transport, and this requires a lot of energy. Unit 3, Cellular Energetics. The substrate, active site, reaction, and products. A substrate binds to the enzyme, and then there's an enzymatic reaction, and the energy required to make chemical reaction varies for the needed substrate and active site. The activation energy required for reaction. There is a biological catalyst, and this can lower the activation energy, shown on the right through the image on the bottom right. And an example is glycolysis, which takes up a lot of energy, and it starts off with glucose and is broken down. The BRCA cell processes that require energy, the formation of ATP to provide for cancerous cells, and the BRCA uses glycolysis to feed cells, so the cells are using up a lot of energy in total, shown through all of these ideas.
Unit 4, Cell Cycle and Cell Communication. Mutations in receptors can prevent proper reception of substrate. Non-functional tumor suppressors lead to uncontrolled growth. For example, tumor repressors P53 and RB1. Overactive oncogenes can lead to tumors, and because oncogenes are like stepping on the gas, it increases the speed of cancer progression. Inherited mutations of the BRCA genes increase potential development, and this is why people who have the disease in the family usually get tested for it. And lastly, in breast cancer, often cell cycle checkpoints do not function properly. Cyclins and cyclin-dependent kinases. Unit 5, Heredity, Chromosomes and gamete cell linkage, sex-linked traits, X-linked and Y-linked. Chromosomes and somatic cells linkage, autosomal dominant, autosomal recessive. Punnett squares made by Reginald Punnett are used to determine the alleles of offspring. BRCA parent alleles cross and they predict outcomes of children, autosomal dominant, and contains one dominant allele. Unit 6, Gene Expression and Regulation. DNA polymerase involved with DNA replication proofreads the newly made DNA and replaces any incorrect nucleotides. DNA sequence passed on to the next generation may become permanent and could be a mutation, which causes genetic variation for the better or worse. DNA replication occurs 50 base pairs per second and 1 month per chromosome. BRCA can spread rapidly through DNA replication. No enzymes stopping cancer spread, mutation within gene, uncontrollable transcription factors are all different variables to cancer.
Unit 7, Natural Selection. The gene flow between different populations of a species. This can create an exchange of alleles, which creates variation. This holds the populations together. And then new species evolve, yet inhabiting ancestral region. This is also known as sympatric speciation. And a blockage could be habitat differentiation. And a topic is microevolution, which is change in allele frequencies. And this can be natural selection, sexual selection, genetic drift, gene flow, which all build toward the macroevolution. And the BRCA mutation is passed on and exchanged, and this can be prominent in some populations, leading to a high frequency of the allele in the gene pool, which could be shown in the right. So population B would have a very high frequency of the red allele, like red butterfly allele. And this mutation can evolve in the same habitat over time.
Unit 8, Ecology. A learned behavior, associative learning, the ability to associate one's environment feature from another, like on the right, the meerkats learning from their parents. Selfless behavior related to population, altruism. So, selfless behavior reduces individuals' fitness because they're sacrificing themselves, increases population's fitness, which drives evolution and chooses not to have children. Exponential growth within population. It's a J curve as shown on the bottom right, and there's no limiting factor. BRCA cancer cells with no outside factors can continually grow, population size increasing.
And my bioethics concern is that BRCA tested individuals can be concerned about their privacy, such as the genetic information, and free information could lead to different consequences. It could be seen as healthcare providers breaking their obligation to keep information private and their own opinion on protecting personal information, the individual. And this can also be seen as genetic information being taken advantage of, such as companies using such data to control the individual's health aspects like insurance, health insurance, and then financial issues due to disability revealed. This can lead to more issues since their information has been compromised.
For my bioethics slide, I did an individual with the BRCA mutation and how it questions family genetics, and it shows the risk factors for others in that family. Familial issues with the family, so that sharing the information about the disease can create tension and stress. Family has to accept the information about the financial costs and the stress, and more issues can follow from testing than without testing. Moral obligations to relatives, the need to tell family and relatives that you are a carrier or have the disease, requirement for tested positive individual, and moral beliefs in testing.
Thank you for listening.
Thank you.